links for the first part of Protein Lecture 1 and then the rest of today. Any questions from last time? So far so good on protein? Yes?
What makes a protein branch again, or an amino acid branch?
It's the actual molecular structure of their side chain. There's kind of a branch point in there. Yeah. So there's only 3 of them that have that. And they all happen to be essential. I'm guessing that structure can't be completed by enzymes we have, so that's why they're all essential. Right here, right? We said that most protein is actually absorbed as di-and-tripeptides. And we said there's a transporter for that. That's called peptide transporter 1. And it transports amino acids across that brush border membrane. So here's another important point of all of this. In general, amino acids need transporters to go across cell membranes everywhere in the body. So amino acids need transporters to go across cell membranes. So even though we're just looking at intestinal cells, when these amino acids go throughout the blood and through tissues, they need transporters to go in and out. And so some of the transporters that are here, the single amino acid transporters, are in other tissues as well. But we're going to stay really general on that because there's so many of them, it gets confusing. So just remember that basic principle I just said. Now, even though a lot of peptides go into an intestinal cell, there are enzymes, there are peptidases inside that cell that cleave them. So even though they go in as di- and tripeptides, they're almost immediately converted to single amino acids. And that's how they can be used. So intestinal cells can use the amino acids that they absorb, or they can allow them to go through the brush border membrane and enter the portal vein. So as these amino acids go in, some are used for things like protein synthesis to make apolipoproteins, new digestive enzymes like maybe enteropeptidase, hormones like CCK, for example, nitrogen-containing compounds, maybe DNA and RNA and things like that. There's a unique amino acid that I want you guys to remember for enterocyte metabolism, and that's glutamine. Have you ever heard of glutamine? Sometimes you hear of it, and I think that one of the reasons it's unique is glutamine side chain has 2 nitrogens. So it can be a good source of nitrogen. But glutamine is it an essential amino acid? It is not. Okay, so we can make it. So it's important but not essential. And when you guys go through clinical nutrition, you might talk about glutamine in some enteral formulas because sometimes it's really important for the health of the enterocytes. And here's one of the reasons why. So here's 2 important characteristics of glutamine. One is it's an energy source. And here's why. Glutamine can be converted to glutamate, and that can be converted to alpha-ketoglutarate. And where's that from? That's in the Krebs cycle. So these enterocytes can pull off nitrogens and eventually get to an energy source. So that's one of the important functions of glutamine in enterocytes. A second important function is it stimulates growth of enterocytes. So it's an important part of enterocyte metabolism. So any amino acids that aren't used in the intestinal cell goes across the brush border membrane into the portal vein and then starts being distributed. So they'll first go through the liver. They don't need carriers or anything like that. They're free in the blood and they'll head towards the liver. So anytime we eat a protein containing meal, this process occurs, so the liver gets an influx of amino acids relatively quickly. And this is where it gets interesting for a lot of reasons. This is the liver metabolism of amino acids. It's the major site of amino acid uptake following dietary intake. So most of the amino acids go into the liver. So what does the liver do with the newly arrived amino acids? We're going to talk about 3 bullet points. You can see, though, these add up to 100%. So we're going to count for all of them in these 3 categories. About 20% of the amino acids that you just ate are used for making proteins and nitrogen-containing compounds in the liver. Now, not all of the proteins that are made in the liver stay in the liver, but the liver does make them. So the liver makes significant amounts of proteins for its own use, and some of them go into the blood, like albumin is the classic example, right? Look at this one. This one's kind of weird. I think that's kind of, okay, that makes sense. 57% of the amino acids are catabolized. That means they're broken down. Remember last time I said they're converted into, when they're catabolized, it's nitrogen, or what do we call the other one? Carbon skeleton. So if we have an amino acid that's catabolized, that means the nitrogen is separated from the carbons of the molecule. And we call that remaining molecule a carbon skeleton. I'll draw that out in a second. Does that seem like a lot? You just ate that protein. Why do we catabolize it? I'll give you some ideas of why that's important and how that gives us metabolic flexibility. 23% of the amino acids are released back into the blood without being metabolized. In other words, glutamate can go in, the liver cell comes back out and goes general circulation. Okay? So essentially you could look at this like, well, the liver's releasing 23% of the amino acids out using them. Kind of like glucose and some of the other molecules we've talked about, but only 23% of them. We'll talk about fractions of that coming up. So let's start looking at each of these bullet points in a little bit more detail. So this is kind of bullet point number 1. This is the synthesis of proteins and nitrogen-containing compounds within hepatocytes. So we have, for example, plasma proteins can be made, like albumin. Enzymes are made. Some of the amino acids convert to each other. I'll show you that in a second. Remember, it's an important source of nitrogen, so the liver can break apart the amino acid and use the nitrogen to make things like nucleic acids. So we don't get a lot of nucleic acids in our diet. We take the nitrogen from amino acids and then make nucleic acids. And then creatine is another good example of this. What's creatine famous for? What's that? Yeah, water retention. Yeah, it causes that. You guys have all heard of creatine as a supplement. How many of you guys have gotten questions about that? Okay. It tends to be a very popular supplement. It's been around for a long time. Relatively safe. Can be effective. What kind of people take it, and why do they take it? Yeah, like bodybuilders, people who try to add muscle. Does it help them? What do you tell them when they say, hey, should I take creatine? Like, no. What do you think? Yeah. Is it safe? Moderation. Right? They're taking it for a couple of reasons. One is a potential energy source. And here's why. So we actually make creatine. Or you can take it in supplemental form. So here's creatine, here's its structure, right? It's got 3 nitrogen atoms. Here's creatine phosphate, also known as phosphocreatine. Look at the difference. Creatine can be phosphorylated. And sorry, these lines disappeared. These aren't floating. This didn't disintegrate. This is actually together. It's been phosphorylated. Where'd the phosphate come from? ATP. So on the slide, next to this, put at rest. At rest. Right here. Because when a person's resting, we can make ATP using, for example, Krebs and ETC. Take some of that ATP and add it to creatine to get creatine phosphate. And here's why. Because during exercise, this is what happens. That phosphate is removed. Phosphocreatine becomes creatine. Where does the phosphate go? It can be used to regenerate ATP. So creatine phosphate is an anaerobic energy source that's important during what kind of exercise? High or low intensity? High intensity, I said anaerobic. During high intensity, long duration or short duration? Very short duration, we're going to say exercise, but basically it's work. Right, remember that in other classes you talked about the energy systems. We have the creatine phosphate, you got the lactic acid system, you got Krebs and ETC and those. Remember, creatine phosphate lasts typically 5 to 10 seconds of high intensity movement. So it's only important for certain types of exercise. There's actually more creatine phosphate in our muscle cells than there is ATP. ATP is labile, doesn't stay around very long, tends to be used rapidly, so there tends to be more creatine phosphate in a muscle cell than ATP at rest. So here's what's happening. Let's take an example. Let's say a person starts taking creatine, and you'll typically take creatine monohydrate, but if you want to spend more money, there's other options. But that's most common one that's been used over and over again. If a person consumes that, one of the questions is, does that increase the amount of creatine phosphate in muscle fibers? For the most part, we can say yes. So we're taking exogenous creatine, it makes it to the muscle fibers, and then it can be phosphorylated. So during rest, we accumulated creatine phosphate. During high intensity exercise, let's say a person's doing, let's say they're CrossFit or whatever, and they're doing high intensity, short duration exercise. Let's say they're doing repetitions with heavy weights. You guys have all done this, right? Let's say, for example, you do 10 repetitions with heavy weights. Why do you stop? Why don't you keep going? Because you're fatigued, right? Muscular fatigue. What does that mean? It means my muscle's tired. Why? Physiologically, what's happening? What's one of the things you're running out of? ATP. So you start running out of ATP, and remember that myofibril sliding element theory where it takes ATP to relax a muscle, contract a muscle, basically slide those fibers, move calcium in and out of the sarcoplasmic reticulum. All that requires ATP. So as soon as you do that, the more high intensity exercise you do, the more fast twitch and more overall muscle fibers you recruit in order to do that work. So let's say I'm doing this at 100%. So let's say if I do my 10 repetitions, I couldn't do another one if someone had a gun to my head, because it's that hard and it's physiologically I can't do anymore. You set the weight down, and this is your opportunity or their opportunity to catch up with friends, right? Now they're waiting. They're going, oh, what are you doing Friday night? What are they waiting for? One of the things they're waiting for is an enzyme called creatine kinase, which is reversing this process where we're regenerating creatine phosphate. Because as that person was going through, let's say, for example, they got to 5 repetitions and they're out of ATP. Partway through that set or exercise, this is happening. Some of that phosphocreatine is regenerating ADP as it's formed from that work. So it's an extra energy source. It's kind of a backup energy source. Now you get a few more repetitions like that, and now you set it down and you're like, okay, that was great. How long do you wait? How many of you guys time? Are you guys obsessive compulsive like me? Oh, gotta go. Nice talking. Do you guys time each other in between stuff? How long do you wait in between? Let's say you're doing something. How long do you guys wait? 30 seconds, a minute? Like high intensity stuff. Whatever that is. A sprint. 90 seconds, a minute. Have you ever thought of this? How long should you wait? Is there an association? If you wait longer, can you do more the next set? Or can you keep, let's say you're running sprints, you're running 100-meter sprints. If you wait longer, does your next sprint speed improve? Or let's say I take off right after 30 seconds and I do it again. Have you ever tried stuff like that? That's an important variable in an exercise regimen. It's why are you doing certain things. If you're trying to add lean body mass, and you're trying to stimulate muscle fibers so that they adapt and become more toned or however you want to think about it. One of the things to think about is that time in between sets. Are you trying to get stronger? Are you trying to improve endurance? If a person's trying to add muscle and get stronger, if you wait longer, it allows more muscle fibers to regenerate ATP so you can go back to what you were essentially. So try this if you're bored in the weight room or whatever you're doing, or a sprint. If you go in there, do your 10 repetitions, maximum amount, and then wait 30 seconds and try it again and count. Next time, wait 3 to 5 minutes. You'll probably be able to do the same number of reps you just did because you've allowed those muscle fibers to regenerate ATP and phosphocreatine. So if a person's supplementing with creatine, they're essentially loading up their ability to do that process. So they're kind of accentuating their anaerobic high-intensity energy source. Does it work? You know what I'm saying? It depends. But high intensity, if a person's trying to improve strength, and if they're doing a creatine regimen, and all of their exercise regimen makes sense, and they're taking care of themselves nutritionally and everything, it can work. It can be effective. And one of the reasons is because it adds more creatine to muscle. And what else does it do to that muscle? This is charged. Draws water into the muscle. In general, a hydrated muscle performs better than a dehydrated one. So what does that mean as far as body weight? Probably going to gain body weight. So one of the things to think about is, so if I started a creatine regimen today, and probably about a week later, I'll probably gain 8 to 10 pounds. And if I'm trying to add muscle, I'm pretty excited about that. Then I stop the creatine. Do I lose all the muscle? Not really. But the first part of it is you gain weight, and depending on what the person wants to do, sometimes that's good, sometimes that's bad. If they're strength athletes and there's no weight classes or categories, then that's not a problem. They typically perform at a higher level and higher intensity exercise movements. If it's a person that is in a weight class, wrestling or whatever, you gotta watch that. Because a person will gain weight. You should consume more water. So a person should consume more water when they're on this. it can, so it can lead to water accumulation. It can improve strength. Because now, if you think about it, now the next set, maybe I can do as many or more repetitions. If I didn't have that additional creatine, I'm not doing as many. In general, there's an association between strength and cross-sectional area of a muscle fiber or a muscle group. So it can be effective for that. So for strength athletes, it can be good if weight's not an issue. How long should a person be on it? So erring on the side of caution here. The problem is and I'm going to say guys, because a lot of times guys don't want to go off it because they don't want to lose the weight. Because then they feel like, oh, I've lost all my strength. Probably not. But a lot of times they'll stay on it for long periods of time. And I'll say, how long have you been on it? Can't remember when they were off it. They want to be off it at least as long as they're on it. So you kind of want to go through cycles of being on and off for safety. Probably maybe a 12-week regimen might be appropriate, and then maybe they're off it for 16 weeks at least. Now for the NC2A, so for college athletes, it's not banned, but it's prohibited. In other words, I should not say, everyone should go on creatine phosphate. If they're on it, they're not going to be tested for it. It's a natural compound. So relatively safe, but I always say be careful. In the past, I've known at least a few athletes that have gotten abnormal liver enzyme levels because they were on creatine so long, which means it was causing issues in the liver. They went off it, stayed off it for months, and things kind of cleared up. So I always say, if you don't need to be on it, absolutely don't be on it.
When you go off it, is it just that water weight that you lose?
No, so when most of the weight loss, yes, it is going to be water, but
do you quickly lose the muscle because you can't maintain that same amount of?
no. So good question. I wouldn't say you quickly do. You might be able to maintain that, or pretty close to it, because the extra work that you've done is what's added that muscle. Yep. Mm-hmm.
How does it harm the liver exactly?
It has to do with the nitrogen metabolism and nitrogen excretion.
Mm-hmm.
Yep. I think that's probably most of it. Yeah.
Would you still gain that water weight even if you weren't working out while competing?
Yes. Not as much. So one of the things to keep in mind is probably 95% of creatine in our body is in muscle fibers. Creatine is made mostly in the liver. Creatine's made in kidneys and liver, kind of a two-step process. And then it's exported into the blood, and it ends up in muscle fibers. So about 95% of creatine in the body is in muscle fibers. And you'll see there's also some other applications for creatine. So creatine is going to be something in nutrition and supplements for a long time, I would say.
You need to drink more water because the water that was in your body is being stored mainly in the muscle after.
I'd say there's two reasons for the water. One of it is to allow the muscle to have adequate access to water, and so that the muscle is not drawing water from other body compartments. So you're not kind of dehydrating some cell types while you're hydrating muscle. So you want to make sure. Second reason is because of that nitrogen, you're flushing nitrogen. So you want water, because we'll talk about urea metabolism in a minute. But it's kind of a safety issue as well.
In general, how much more water?
It depends on the person. Boy, I'd probably say 25-30% more than they typically drink. Depends on body size. Percentage is a decent way to look at it. So, and the other thing that I want to mention, one more thing. For safety issues, a person doesn't need to do the saturation phase. Supplement companies love to say, hey, for the first 5 to 7 days, consume 20 to 25 grams of creatine. That's a lot. You're essentially mixing it in and drinking it a few times a day. Doesn't need to do that. Lots of studies now have shown if you don't do the saturation phase and you just use, for example, 3 to 5 grams of creatine a day for a relatively large athlete, it takes a little bit longer to get the effects, but it's a better way to do it. Okay, so no saturation phase on that. I'm not promoting its use. I just think you guys get a lot of questions about that. And so I wanted to spend a few minutes on that. We good there? Any other questions? And it tastes great, I should add. All right, here we go. You can imagine it's like chalk. This one looks simple. Oh, this is great. That's some of the things the liver can do. Let's just look at the main points of this. So we're talking about that first bullet point. We're talking about the nitrogen-containing molecules being made. So this is what the liver can do with some of these. For example, histidine is converted to other molecules. So each of the molecules in these boxes obviously is an amino acid. The other ones up here, almost all of them are nitrogen-containing compounds. So the amino acids are converted to other nitrogen-containing compounds. And there are tons of them. So we'll talk about a few of them as we go through. But here's another important point. The liver can take essential amino acids, convert them to non-essential. So there's lots of amino acid interconversions. The liver can take non-essentials, convert them to other non-essentials. What can't we do? You can't go non-essential to essential. But what if you have an amino acid that you can normally make, and yet a condition occurs that you can't make that non-essential amino acid? It then becomes essential. And that's what this is talking about. So PKU stands for phenylketonuria. In other words, phenylalanine is converted to ketones and it ends up in fluids, including urine. And that's known as PKU. Have you heard that term? It's an inborn error of metabolism. Inborn error of metabolism, what does that mean? It's genetic. It's a genetic alteration, typically of an enzyme that alters a metabolic pathway. So it's a genetically modified enzyme that now changes the metabolic pathway. We can call those inborn errors of metabolism. And there's tons of them. PKU happens to be one of the more common ones in the U.S. I think last time I looked it up, the incidence is about, so it's autosomal recessive, the incidence is about 1 in 15,000, I think, in a typical U.S. population. Obviously, it runs along ethnicity sometimes, but
Could you please repeat that sentence? Genetically
modified enzyme that then changes the metabolic pathway. Is that right? So here's what happens. Typically, phenylalanine can be converted to, give you a hint. Oops. Draw the structure wrong. So here's a side chain of phenylalanine. This side chain of tyrosine. Phenylalanine is essential. Tyrosine is not. Why? It can be made from phenylalanine, typically. In individuals with PKU, they have an alteration in the genetic sequence of an enzyme called, what would you call this enzyme that adds this? That's a hydroxylase. More specifically, it is phenylalanine hydroxylase. There's a few common alterations in the sequence of the gene for this, so the protein structure is modified and it's not effective. So in that case, what's happening in the liver? What's happening in phenylalanine levels in general? They going up or down? They tend to go up. Why? It's not being converted. So phenylalanine tends to go up. And what happens to tyrosine? It's typically low. Because this enzyme is not converting it, phenylalanine accumulates and is modified to form phenylalanine ketones. And these tend to be harmful, especially to the brain and central nervous system. So typically one of the first screening tools, I think it's, oh no, I'm trying to remember. They always screen for it, but I don't know in California if it's required by law. I don't think it is. It is required by law in California? Okay, everyone does it, so it might be. If a person is diagnosed with PKU immediately, that early on, what would you do? What's the nutrition intervention? Do you eliminate phenylalanine? Why or why not? Who says eliminate it? Okay, who says no? Keep giving it to them. I think it was about 3-2. You don't want that to accumulate, but you still have to have phenylalanine for protein synthesis. So you control phenylalanine intake. Let's say you moderate phenylalanine intake. What do you do to tyrosine? Increase it. Increase tyrosine. How would you do that in a newborn? Yeah, synthetic diet, synthetic formula, specifically for PKU individuals. And as long as that happens and they're screened and they control that right away, they'll do blood tests consistently for years, but most of the time they're normal life. They can control that. The phenylalanine doesn't accumulate and they're fine. A few years ago we had a nutrition student, her brother had PKU. So there's probably 1 or 2 Cal Poly students on campus that have PKU if you think about the numbers. Okay? Later on in life, what a person will tend to do is they'll tend to move towards diets that have lower animal proteins and higher plant-based proteins, because animal proteins tend to have higher amounts of phenylalanine, so that you can kind of control it that way as well. Different levels of severity and everything, but that's a common example of, in that case, tyrosine, which is normally non-essential, becomes essential in the diet because of the condition of PKU. So tyrosine is the conditionally essential amino acid. So you can look at some of the other ones here. Now, so here's the phenylalanine tyrosine. And then tyrosine, remember that's a lot important for a lot of neurotransmitters and everything. So you can see in some of these, like methionine, essential. Cysteine, non-essential. Why? Because we make it from an essential. And then some of them has to do with non-essentials converting. So the second part of this. Almost 60% of the amino acids that go into the liver are broken down. So you end up with the amine group and the carbon skeleton. So the liver is the main site of catabolism for most of the amino acids. Not the major site of the catabolism of the branched chain amino acids, though. We'll talk about that in a second. That occurs in the muscle. So branched chains have the unique aspect of they're metabolized in muscle cells, not the liver. Liver uses branched chains for protein synthesis, but doesn't metabolize them into other molecules in general. And I'll tell you why in a few minutes. So that's another reason why sometimes we'll say, person exercises a lot, they should supplement with branched chain amino acids. What's the most popular protein supplement, would you say? Whey protein. Whey's high in branched chain amino acids. That's considered a good thing because all the branched chains are essential. And leucine tends to be anabolic. So that's one of the reasons whey protein can be effective as a protein supplement. I'm biased. I like whey protein. I've seen it work a lot of times. It's not for most people, but it can be effective, and that's one of the reasons. So what happens during catabolism? In other words, when this amino acid is broken down, what's happening? Well, you're generating a nitrogen-containing molecule that's no longer part of an amino acid anymore. So mammals have to deal with that nitrogen. Accumulation of nitrogen in mammals is toxic. We all remember that from probably other classes, right? The nitrogen has to remove from the amino acids by one of two major reactions. One are called transamination reactions, and the other one is deamination reactions. We'll look at that in a second. The nitrogen after deamination, or if that nitrogen becomes free, can be incorporated into the urea cycle. We'll look at that in a second. That's what protects us. What happens to the amino acid carbon skeleton after nitrogen has been removed? Well, it depends. If a person has a lot of calories, including too much protein, they can take that carbon and make fat. If they are calorically challenged or low in glucose, they can take those carbon skeletons and make glucose or energy. So we'll look at a diagram in a second. Here's why some of these amino acids are non-essential and some of them are essential. And we're essentially here. We're going to look at transamination reactions. So if we look at this example, here's alanine. We've talked about it a lot. Alanine can easily be converted to pyruvate. The only difference in their structure is an amine. Aspartic acid can be converted to oxaloacetate. Glutamate to alpha-ketoglutarate. We saw that one a second ago. Okay? So there's lots of interactions here. The difference, the nitrogen. So here's an example of an enzyme that converts keto acids to amino acids back and forth without the nitrogen being released. So here's pyruvate. Here's alanine. Only difference is add an amine group. Where does the amine come from? Typically glutamate. When that amine group comes off of glutamate, you end up with alpha-ketoglutarate. So the amine group has been transferred. It was never free in solution. So this is a transferase reaction, an aminotransferase reaction, or a transamination reaction. It's transferred, not free. That's why this enzyme is called alanine aminotransferase. So this is kind of a weird reaction. Look at what's happening. This enzyme will tend to, let's say it's going forward. Let's say we have glutamate. The enzyme will hold on to the amino acid and pull this nitrogen off and essentially hold on to the nitrogen and transfer it onto a keto acid. So it'll transfer it onto pyruvate. So the amine group is never let go. So in this case, there's kind of two substrates and two products. So you can probably look at it like this, I guess. Let's see. Here's the enzyme. Let's run it this way. Let's say this is alanine. Here's alanine. Red pen, obviously, right? Okay. So what the enzyme can do is, if it pulls this amine group off, now alanine, this is now what? Pyruvate. Here's a second keto acid. This is alpha-ketoglutarate. So what the enzyme does is move the amine group to this. And alpha-ketoglutarate becomes glutamate. What's that? Yeah, it'll go either way. So this is bidirectional. Those arrows don't show it, but this enzyme can run that either way. And this is a very common liver enzyme. Have you guys ever looked at blood panels or chemistry from a person's blood work? Have you ever seen that abbreviation, ALT? Or sometimes they'll go ALTase. You look at it and go, okay, normal. Why are you looking at it? You're looking for liver dysfunction. This enzyme is very common in the liver, not in the blood. So if the liver is being damaged, this enzyme will go into the blood. And you'll see that on a test. If you see that on a person's blood work, that means some of the liver cells are being damaged, and these enzymes are leaking into the blood. There's a couple of these transaminases that you'll see in clinical nutrition when you're looking for things like liver dysfunction. You're looking at those enzyme levels. Okay? So you want to remember that. And again, this is a transamination reaction. Here's another reaction. These are called deamination reactions. This is when the amine group is actually just removed, as that term implies. So in this case, here we have threonine. The amine group comes off and goes into solution. It's not transferred onto a onto a keto acid. So you end up with a keto acid and an ammonia, which in our body, ammonia and ammonium ion go back and forth. So ammonium ion is NH4. Ammonia is NH3. Both of them are harmful. So our body tries to purge ammonia and tries to minimize its production. I'll tell you how in a second. Do you have a question, Emily?
Yeah, is the enzyme, is the name of the enzyme changed depending on what the product is? Like, will it always be alanine, or if we're making something else, would that change?
Good question. That same enzyme name can do quite a few. It actually is relatively, it's not very picky. It can work with a few amino acids and keto acids, but it's typically called that, alanine. Yep. Depending on the cell needs, depends on direction and also substrate. So the cell has kind of regulatory mechanisms for that. Okay. So here's one of the important things that mammals have to be able to do, and that's make urea. So here's the urea cycle. So a few kind of big picture things. Urea cycle is in the liver. It helps us control ammonia levels. I think we all know ammonia is harmful to us, right? So it helps us control that by making urea. So here's how it works. We look at the liver, right? Obviously exposed to a massive blood supply going in and out. If ammonia is in the blood, that ammonia can run through the liver, and the liver can take that free ammonia and add it into urea. So that's one of the things that's happening here, and I'll show you how that works in a second. So the urea cycle converts it to urea, and then the urea leaves the liver, goes into the blood, and travels to the kidneys, and the kidneys excrete it. So urea ends up in the urine. So urea is a part of urine. And that's what's happening here. Now that ammonia could come from the blood, or that ammonia could be happening in deamination reactions in the liver. So the liver itself might make some ammonia, but then in that case it would immediately go into the urea cycle. So this has to do with nitrogen metabolism. Have you ever heard anybody say, well, eating a high-protein diet helps a person lose weight because it increases our energy expenditure? You ever heard things like that? Urea cycle takes energy. If a person eats more protein, urea cycle activity goes up. That's one of the things they're leaning on. Absolutely, that's the case. If everything is working appropriately, we should be able to modify urea cycle activity to dietary protein intake. And I'll tell you thresholds in a minute. But this is how it works. And there's only a few important points that I want you to take from this. If you memorize this, that's just for you. Okay. Now, sorry. Here's the first reaction or first step in the urea cycle. Here's ammonia. Ammonia combines with CO2, readily available, right, from Krebs cycle and lots of other reactions. These two combine to make carbamyl phosphate. So here's the nitrogen, here's the CO2, phosphate's added. It takes 2 ATP to do that. So it requires energy to start the urea cycle. Once you have the carbonyl phosphate, it combines with ornithine. And you might have heard of some of these molecules, right? These are amino acids. When those combine, you end up with citrulline. Relatively large molecule. We're going to track this nitrogen that came in from ammonia. Now it's here. Citrulline combines with aspartic acid, another amino acid. So here comes another nitrogen. Now look where it's added. Here's aspartic acid. It's added right here. So the nitrogen is now bound to this carbon. That molecule is called, appropriately probably, argininosuccinate. That splits into fumarate and arginine. Have you heard of arginine? In some species, that's an essential amino acid. It's important for growth and nitric oxide metabolism and things like that, but it's part of the urea cycle. Now we have a nitrogen atom here and one here. Now look what happens. Urea splits off. We get back to ornithine. So how many nitrogens does urea have? 2. So one important point of this is we're going to say in one cycle of the urea, in one turn of the urea cycle, you can eliminate 2 nitrogens. So one turn of the urea cycle eliminates 2 nitrogens. How many ATP does it take? That's AMP. 4. So each urea molecule requires 4 ATP to make it. Two here. And 2 ATP equivalents here. So that's 4. And then the liver cell will put urea into the blood. Urea is water-soluble, non-toxic. It's so water-soluble. Back in my protein purification days in the lab, we used to use urea basically to bring proteins out of solution. It does very well in water. It's very soluble. But then that ends up in the urine, and then it's purged. So you've gotten rid of that ammonia. You've controlled that. But it's important that it's working properly. If a person has liver dysfunction and the urea cycle is not working properly, you have to minimize protein intake. So liver dysfunction requires, let's say, dietary protein adjustments. What about the kidney? Same thing. If there's kidney dysfunction, you also have to moderate protein intake because it's important. It's got to be able to excrete the urea. And we'll look at this in a second, but there's also ammonia in the urine. So if kidney dysfunction is a problem, ammonia is not being purged as well. Okay, so those are some of the important points on this. Let me mention this now. I think I did this before, but remember, I'd say high protein intakes in general doesn't cause liver dysfunction or kidney dysfunction. But if either of those are present, then you definitely control protein intake. And before, I think we said, probably 300 grams is a ceiling for protein, which basically is more than enough for just about anyone. So most people don't get close to that and shouldn't. Even really people that are trying to add a lot of lean body mass and they are big in size, typically like maybe 225 grams of protein is typically enough. And we'll talk about that in a second. Let's take a break here. Huh? So let's take a quick break and then we'll finish this and do protein lecture 2.
Can I still turn this on the homework assignment for today? Or the extra credit assignment?
So I'm going to adjust that score. So let's finish up this one real quick. This gives you an idea of the importance of the urea cycle. So if you look at the major forms of nitrogen excreted in the urine, urea is by far the most important one. So this is the most important route of nitrogen excretion for us. So the urea cycle is very important for controlling that. You can see if you add up all the nitrogen and percentage-wise, it's about 85% of the nitrogen in the urine is as urea. There's some ammonia. Sorry, that's wrong, right? That should be NH3 or ammonium ion. But there's ammonia in the urine too, a little bit. Creatinine, where does that come from? What does that remind you of? Creatine. So in muscles, creatine is converted to creatinine, and then creatinine is excreted through the urine. And then uric acid, what's that famous for? Associated with gout. So that's a normal nitrogen-containing compound in the urine. When that accumulates, it tends to cause an immune response, and that can cause pain in joints. Now, glutamine. I mentioned glutamine before. I said it was unique in enterocytes because it's an energy source and it stimulates growth. But glutamine is also important because it is kind of shuttling nitrogen to other tissues. So the liver can deal with ammonia and just incorporate it into urea, but other tissues are also turning over amino acids and they're ending up with free nitrogen or ammonia. And what they can do is they can add it to glutamate. So let's say this is a muscle cell and it's degrading amino acids and it's ending up with some ammonia through deamination reactions. There's an enzyme called glutamine synthetase that'll take that ammonia, add it to glutamate to give you glutamine, and then glutamine goes into the blood. So if you measure glutamine in the blood at any time, it's higher than probably most other amino acids because it's shuttling nitrogen. So rather than making ammonia, cells can make glutamine to move nitrogen to other tissues like the liver. Okay? So one of the other things to think about is on those amino acids is when the amino acid is broken down and you have that carbon skeleton, there's a few different options for that. So I would commit this diagram to memory, but then also understand its relevance in certain conditions. So if we look at this diagram, here's an amino acid. And again, in this case, the nitrogen, the amine group is being pulled off, and now you have the carbon fraction left. So it has possibilities here that we'll talk about in a second. But that amine group can be used to make non-essential amino acids. Not essential, so be careful here. Non-essential amino acids and other nitrogen-containing molecules. Or It can be incorporated into urea, or it can stay as ammonia and go into the blood. And both urea and ammonia are excreted by the kidney. The carbon skeleton, its fate is kind of determined by the needs of kind of the cells and the overall system. Let's say a person is inadequate in energy intake, that we can take those carbon skeletons and use them for energy. In this case, what does that CO2 mean? That means Krebs cycle, right? That's respiration. So the amino acid carbon skeleton is going into Krebs cycle to provide energy. In inadequate calories or inadequate glucose, then glucose can be made from the carbon skeletons. Or ketones can be made from the carbon skeletons. Almost all of the amino acids are gluconeogenic. Most of them are ketogenic. So we make ketones out of some of them too. Okay, so in that case, it's an energy source and a glucose source. Fatty acids are the opposite. So if I ask you to describe what happens during starvation, don't say fatty acids are going to be made. Because in starvation, we're getting energy from it, glucose and ketones, not fatty acids. In excess calories, including too much protein, you can end up with fatty acid production. All right, so the scenario there is person's consuming a lot of calories, including a lot of protein, above demands. Some of those carbon skeletons make fat. And most of that's going to happen in the liver. So here's the role of branched chain amino acids. So they did this experiment years ago, and they took animal tissues and they gave these animals mixed diets. And in this diet, the protein source was about 20% branched chain amino acids. So when they looked in the portal vein and they got a ratio, they did amino acid profiling. Then they took blood samples coming out of the liver. And they found that coming out of the liver, 70% of the amino acids are branched chains. In other words, branched chains are not metabolized much at all in the liver, but the other amino acids are. So you kind of get an enrichment effect. That's when they first realized branched chain amino acids are more important outside the liver, and then they realized it's actually in muscle. So branched chain amino acids have unique roles in muscle compared to the other ones. All amino acids obviously are used for protein synthesis in muscle, but muscles also use branched chains for energy. Isoleucine, leucine, and valine, the three branched chain amino acids. We're going to simplify this. You might talk about more about this in 329, hopefully when you talk about this enzyme. The muscle cells can take these branched chains and make Krebs cycle molecules. So they can get energy from them under what conditions? Aerobic or anaerobic? Aerobic. So a highly trained person that's done a consistent aerobic training kind of accumulates the ability to use branched chain amino acids as more of an energy source. So it can become more of a significant energy source for them. It's never close to what carbohydrates and fat is, but it does occur. And then during this process, these muscle cells might be making ammonia, and so they'll make alanine and glutamine and put those into the blood to control nitrogen levels. Okay, so that's one of the reasons they're unique. So let's go to protein lecture 2. So a big part of this lecture will go pretty quick. A big part of this lecture is, thanks for not clapping, is how much protein do we need? That's what we're going to talk about here, and there's a few ways to do that. So we're going to talk about protein needs and nitrogen metabolism again, then we're going to talk about protein quality. So how much protein and what types of proteins are appropriate under certain conditions. So how much protein do we need to eat each day? So this is a clue to that. if a person's not trying to add lean body mass, is not growing, is not pregnant, or any conditions where they're trying to add more tissues or anything like that or recover, so normal adults that have no significant health conditions or demands, we need to eat the amount of protein that we lose. We just need to replace losses. So that's why this is a clue. Replace losses. How do we lose protein? We can lose protein in things like lose skin cells, lose hair, lose fingernails. Sweat contains some protein. Lose body fluids. But we tend to lose more nitrogen containing molecules than protein. Sure. We tend to lose more nitrogen containing molecules than protein. So here's the principle. Protein goes in, nitrogen comes out. So we tend to eat dietary protein to get nitrogen. We tend to lose that protein as nitrogen, not protein. Remember urea? That's the major route of nitrogen excretion. It's not a protein, it's a nitrogen containing molecule. Where'd the nitrogen in that urea come from? Dietary protein. So the game is you kind of determine how much dietary protein needs by calculating and measuring how much nitrogen they've lost. That's what's called nitrogen balance. And this is just a couple quick slides to kind of set us up for that. So this is estimated daily loss of nitrogen by various routes. And this is out of a sports nutrition text. It's not in your textbook. Key points here. This is daily nitrogen loss, and this is in mg per kilogram of body weight. So don't worry too much about the absolute values, just the relative values of these. Major source of nitrogen loss from the body, urine. That's the major route. What's the form? Urea. Okay, but the route is in the urine. Second major route is in the feces. I'll show you why in a second. Skin losses and then some of the other ones that I always talked about. But you can see most of it's in the urine. Quite a bit of it, though, ends up in the feces. And here's a couple of reasons why. So this shows some of the nitrogen-containing compounds and proteins in the urine. And we talked about this before. Most common one, urea. You don't lose much creatine out of the muscle until after it converts to creatinine. So look at the structure. Here's creatine. Here's creatinine. It's a cyclic structure. So creatine slowly converts to creatinine and then is purged from muscle. So you'll see later on in clinical nutrition, sometimes you'll look at a person's muscle mass or loss of muscle based on creatinine levels in the urine. And if they go up significantly, if they're not adding weight, they're catabolic phase. They're degrading muscle and you're picking up creatinine in the urine. What about amino acids? You see some in the urine. Protein, you shouldn't. Protein in the urine typically indicates kidney dysfunction. So that typically shouldn't happen. Some ammonia we can get rid of in the urine. And then here's uric acid. And a lot of those come from nucleic acid catabolism. And you'll see some of the questions in the study guide are for that slide, so pretty basic there. So we said we need to kind of think about, well, we consume protein, but we lose nitrogen, so how do we compare? Simple calculation. If I have 100 grams of pure protein right here, and I kind of pile that in with a scoop, and I weigh it out, it's 100 grams of protein. how much of that is nitrogen? 16 grams, approximately. And the reason it's approximate is because, you know, every amino acid has an amine group. Some of them also have nitrogen in their side chain. So it depends on the amino acid composition. But in general, we say 16% of the weight is nitrogen. So if you want to convert grams of protein to grams of nitrogen, you take your grams of protein times 0.16. That gives you 16% of it. And an easy way to remember this is remember the protein number is always higher when you do these calculations, because I'll ask you to do one of these, at least one of them. So to convert nitrogen to protein, take grams of nitrogen, multiply it by 6.25. So basically the inverse. So think about what we're doing. Well, if I measure how much nitrogen, let's say, is in the urine, I can kind of calculate how much dietary protein that represents by doing that. And that's what this shows. So 5 grams of nitrogen in the urine would represent 31 grams of protein consumed. So nitrogen balance studies. So they're used to basically calculate dietary protein needs for a population. So here's a basic definition, and this they just compare. So nitrogen balance is the difference between the amount of nitrogen consumed per day, and the amount of nitrogen excreted per day. And that's represented by these equations. So essentially, nitrogen balance equals the grams of nitrogen consumed minus grams of nitrogen lost. Another way to think about it is intake minus excretion. So if a person's consuming more protein than they're excreting, it's a positive number. So we say that's positive nitrogen balance. If for some reason they're excreting more than they're consuming, they're in negative nitrogen balance. Okay? So let's look at kind of a graphic of this. So the core need a little scale up there, right? Intake versus excretion. If intake is more significant, then that's positive, and vice versa. So we look at dietary protein coming in, right? It's exposed to amino acid metabolism in the liver and other tissues. Some proteins are made from that dietary protein. They're degraded. Some of the amino acids are reused. Remember all that? But that can be a way it's being retained, but we don't have to pay attention to that here. We're interested in intake minus excretion. Some of that protein is basically lost as, let's say, enzymes or something that go into the small intestine and they're not reabsorbed, so that it ends up in the feces. So some of the nitrogen in the feces is from proteins and cells that were from our body that end up in the feces. Do we have to replace it? Yes. Okay. But if we look at all of that, there's kind of this cycle that we can think about. And any unabsorbed amino acids that are in the gut, they end up in the feces as well. And I'll give you some percentages for that too. So let's talk about positive nitrogen balance. Is that a good thing? Consuming more protein than you're excreting, essentially. It depends. If a person's growing, adolescence or anything like that, or even if a person's trying to add lean body mass, trying to add muscle, you want to be slightly positive in nitrogen balance. Pregnancy, recovery stage after illness or starvation, you want to be positive. And then the last one I just mentioned. What about negative? In general, not a good scenario, but sometimes people are in negative nitrogen balance. Let's say they consume inadequate amounts of protein or inadequate energy intake. if they're not consuming adequate amounts of calories, then some of those amino acids are used to provide energy, and that compromises their nitrogen metabolism. They can end up in negative nitrogen balance because of that. Deficiency of essential amino acids. And we'll talk about protein quality in a minute, but a high quality protein has high amounts of essential amino acids. If a person's not consuming that, then that can compromise protein synthesis and amino acids are degraded. If there's increased protein loss, and maybe this is because of an illness, let's say kidney dysfunction or something, that can cause protein losses. Or bed rest. I mentioned this before, but when we talked about protein turnover, as soon as a person's not stimulating skeletal muscle, we tend to get rid of that skeletal muscle as an adaptation to our scenario. So as soon as a person becomes bedridden, nitrogen metabolism changes significantly, and you have to be careful or they'll go into negative nitrogen balance. Giving them more protein, a lot of times isn't going to fix that. You have to stop the catabolic phase by some mechanism. Nitrogen equilibrium. This is when protein intake equals protein losses. Who wants to be in this? Most adults. We should be pretty close to nitrogen equilibrium. So that we're kind of replacing losses. We're consuming the amount of protein that we need based on our nitrogen metabolism. How many guys have participated in a nitrogen balance experiment? Well, how do we know how much protein we should eat? I'm thinking about doing this next. I don't know when. It probably won't be spring quarter, but maybe next year we're going to try to teach a sports nutrition class, and I'm trying to get a sports nutrition lab on the books. And one of the fun things to do, I think, would be kind of a nitrogen balance experiment. So in order to do this, I had the opportunity to do this in grad school with rats, where we were studying mineral metabolism and how certain deficiencies altered metabolism through the body, including nitrogen metabolism. So we had we had a big rat hotel in the bottom of our study building in our department building, and we had all these clear plexiglass cages that were called metabolic cages, and we had them hooked up to kind of a respiration system where we could activate our system. We could tell exactly how many calories they were burning. We could tell what their fuel sources were based on CO2 and O2 levels, and we ran that 24/7, and then we had them on different diets. And the other thing that we did was we weighed their food every day, took notes, and then weighed our food and recorded the amount that we gave them, so we knew exactly how much nitrogen they were consuming. We also knew how much nitrogen they were losing because all of their urine and all of their fecal material was collected. I gave that to somebody else to analyze for nitrogen. I had that luxury. I was like, you can do this if you want to learn how to do it. I'm like, no, go ahead. Right? So you concentrate it, and then you quantify the nitrogen. So you can do nitrogen in, nitrogen out. And one of the things that we found was some of the minerals significantly compromised lean body mass, and one of the reasons was because these animals went into negative nitrogen balance. Can you do that with people? Nitrogen balance experiments? Absolutely. That's what the RDA is based on. Almost all of the data for the RDA is nitrogen balance experiments. So here's how they work. You show up and you're like, oh, I'm gonna go to this Pennington Institute or UC Davis and participate in this experiment. And you show up and they're like, hey, you're welcome to stay 5 days, 7 days, we'll pay you some money. You're like, sure, I'm on spring break. This is a great spring break. So you go there and you stay 5 days, and they say, okay, this is how much protein we're going to give you. And they give you this tray. It's a metabolic kitchen. They say, hey, there you go. They give you a tray. Everything's weighed. They kind of watch you. See, no, they don't watch you. But you eat the food. That whatever you don't eat, you give them back the tray. They weigh it so they know exactly how much. In this case, you're supposed to eat this much. This is grams of nitrogen. How much protein is that? 12.1 times 6.25. That's about 75 grams. Is that enough? I don't know. Let's find out. That's intake. Now what do we do? Minus excretion. Okay? In the urine, there was 10.4 grams of nitrogen. Fecal material, 0.55. So this is intake minus excretion from the two major routes. Sometimes they just measure urine and estimate feces. It depends. This is more complete, obviously. But if you add those up, intake is 12.1 minus about 11, something like that. That's positive. Intake is greater than excretion. So is that enough? It is for this person. This person stayed for 3 days and went, yeah, I'm kind of done here. Okay, so they have 3 days of data. Is that enough for them? No, they're negative nitrogen balance. And if you look at these other people, so slightly positive, slightly negative. So what are we doing here? We can take a subpopulation, do nitrogen balance experiments on them, average their overall nitrogen balance on a set amount of protein and determine whether or not that amount of protein is an appropriate recommendation for this population. Assuming they're healthy, same age, maybe same gender, things like that. And then we can say for this population, that's an adequate amount of protein. There's a few assumptions there, but that's generally how you do it. So they did lots of these studies, and that's how we came up with the RDA. And I'll give you some more examples coming up. Okay? Now, one of the questions is, okay, I get the intake versus excretion. What if I cut off intake? Is there still excretion? If I say, no more protein tonight after 8 o'clock, I'm going to have dinner. No more protein. Would I continue to excrete nitrogen? Yeah, because nitrogen-containing molecules are still broken down. Would nitrogen excretion decrease? Yes, but it would always remain at some level. You're always losing some, and that loss is called obligatory nitrogen loss. That's the amount of nitrogen lost from the body as a result of the breakdown of the body's own proteins and amino acids. In other words, that's kind of a required or minimal amount of nitrogen loss. And here's how you do those studies. And you can kind of see a couple things from this. In order to calculate obligatory losses, you don't consume protein. So it's all carbohydrate and fat. They consume that, and you can see, for example, day 1, these are their nitrogen losses, relatively significant. And then as they consume less nitrogen, your body kind of makes a few adjustments. First of all, there's none coming in. So it eventually kind of goes down. After a few days, it'll kind of get to a set level. But you're still losing nitrogen. Here's one of the reasons that's important. Let's say I'm trying to provide nutritional support to a population. Let's say it's astronauts or military or somebody in extreme conditions. And let's say I'm going to try to provide them a nutritional support, a minimal amount of nutrients that will help them sustain life. One of the things you got to figure out is how much nitrogen should be in there. Okay? When you do that, you end up with this number. And if you look at this, if you add this up, this is actually it's a little under 4. And this is grams per day. So if you do this math, obligatory nitrogen losses in this case end up being about 24 grams of protein a day. Is that a lot? 24 grams? You guys know how much protein you eat a day? I mean, I would imagine some of you in here probably eat maybe 40, 50 grams of protein, and others quite a bit more. So I look at that, I'm like, that's not a small amount. One of the things that they figured out from this was in the 1930s and 40s when the World War was occurring, they did experiments like this, and they found out that if they add carbohydrate, it cuts nitrogen losses in half. So this nitrogen-containing diet, nitrogen-free diet, contained approximately how many grams of carbohydrate, you think? 150 grams. If they eliminated carbohydrate, nitrogen losses doubled. Very important finding, because carbohydrate has a protein-sparing effect. And why is that? Yeah, because you're not, now you have some carbohydrate, you are minimizing the amount of amino acids that have to be used for glucose. So that's one way to look at it. Another way to look at it is if a person is trying to maximize nitrogen metabolism in their body, like add lean body mass, they need to make sure they have adequate amounts of carbohydrate. If they don't, they're going to take some of the nitrogen and use it to make glucose, and they won't retain as significant gains as if they would if they were optimizing carbohydrate intake. So how much protein do people need? Here's the number we talked about before. 0.8 grams dietary protein per kilogram of healthy body weight. This is assuming a healthy population and healthy body weight. In other words, if a person has a lot of adipose tissue, there's not much nitrogen in that adipose tissue. It doesn't require as much nitrogen to maintain. So that's why it says healthy body weight. But this is the ballpark number, right? This is what we call the RDA. What's RDA stand for? Recommended dietary allowance. Okay? And that's where you come up with the RDA for protein. Remember the RDA calculations? If I say a person weighs 154 pounds, you're going to say, oh, that's 70 kilograms, so now I can go 70 times 0.8, that's 56 grams a day. Remember, this is adequate amounts of energy, healthy population, healthy body weight. Got to have adequate amounts of energy or you change metabolism. What about if the person starts exercising? Increased demands. I'm going to give you two ranges here. Strength training athletes. Recommendations for strength and power athletes, 1.4 to 1.75 or 1.8 grams per kilogram body weight. Is that more than the RDA? Yeah, that's twice. How would you figure out this number? Nitrogen balance experiments. Can you do that with groups of athletes? Absolutely. And that's one of the main ways they come up with that. Why do strength athletes need more? They need more nitrogen to add and maintain lean body mass. Endurance athletes, do they need more? More than the RDA. Remember, these are not RDAs. These are just recommendations. But these are not DRIs. But the recommendation for endurance athletes is 1.2 to 1.6. So more than the RDA, but not as much as strength training athletes. Endurance athletes need more for addition of lean body mass, and an energy source. Remember, they tend to improve their ability to use amino acids for energy. Have you ever seen those numbers before? Some of you might have, but I would say those are the numbers. Let me add one more point. I would say these are ceilings. More protein than that is not beneficial unless they're on some type of performance-enhancing med. They won't be able to assimilate more than this. So I'd say that's a ceiling. So if you're ever working with athletes, you can use that as a ceiling. Make sure you stay below that mark, and then make sure you hit your carbohydrate numbers that are appropriate, and then control your fat intake, and then that's kind of a good ballpark way to start on something like that. It's difficult to meet the RDA. Typically, no. There's some pretty good protein sources. So this is just kind of showing, for example, 3 ounces of chicken or something. Remember 3 ounces? So 3 ounces is kind of like, palm of my hand's probably like 5 ounces. Stack of cards, remember that? 3 ounces, stuff like that. So in general, not that much. Animal sources tend to be more concentrated in proteins than plants. And there's also a quality difference. We'll talk about that right now. Couple terms. Have you heard this? Complete versus incomplete proteins? What's a context you've heard that in? People will say, oh, I think this protein is good because, well, it's a complete protein. Sometimes you'll hear that for like animal-based proteins versus plant proteins, right? And I'm not promoting either one. I think a diet should be a combination of both of those, personally. Everyone kind of has different preferences. My preference is kind of based on some of the amino acid information on them. But I think, too, we find out a lot of people will say, oh, you know, quinoa. I'll pick on quinoa because it's been around for a while. They'll say, oh, quinoa, that's a great protein because it's complete. Lots of proteins are complete. Complete means it has all the essential amino acids in approximately the amount needed by humans. So if you look at this, I'd say complete, incomplete kind of confers the idea of it's yes or no. I think we need to think a little bit more about on a continuum of higher versus lower quality. Okay? So along that line, let's talk about this. Incomplete proteins. They tend to have too little of one or more of the essential amino acids. So we need those essential amino acids. We can't make them. If a protein is low in amino acids, if it's low in essential amino acids, one or more of those essential amino acids can compromise protein metabolism. One way to get around that is to take 2 different sources of protein with varying amino acid profiles, making sure that the low amounts of amino acids in one proteins are complemented by the amino acid profile in others. And that's the classic beans and rice, right? Mixture proteins. And you can see here, for example, legumes are low in methionine and tryptophan, essential amino acids, and grains are the opposite. Do you need to eat these at the same time? A couple studies came out a few years ago that said it's probably better if you do. Closer together. And I think that has to do with the tRNA loading and appropriate tissues that are trying to perform protein synthesis. It's better if they're loaded at the same time. So it's better if they're closer. But another way to think about this is protein quality. So protein quality is based on 2 main criteria. One is the essential amino acid profile. And the second criteria is digestion and absorption. So on the first point, essential amino acids, in general, we are closer to animals genetically than we are to plants. So the essential amino acids that we need tend to be more closely matched with animal proteins than plant proteins. So in other words, a perfect protein is we lost this protein, now I can consume that in the diet and I get the same essential amino acids. So that's one way to look at it. The other one is digestibility. Sure, that's amounts of essential amino acids in a particular protein source, and how closely that matches our requirements. And the second point of that, if you look at digestibility, plant proteins tend to be digested and absorbed at probably around 85%, 80 to 85%, where animal proteins are probably 85 to 95%. So digestibility and absorption tends to be higher for animal proteins. So one of the questions is, can you just eat more plant proteins to compensate for that? Theoretically, yeah. But you gotta keep that person should keep that in mind. So the RDA, what does it assume? A mix. The RDA assumes the protein intake is from a mix of dietary protein sources, both animal and plant. And you can actually show this if you took two groups of rats that are growing and you put one of the groups on an inferior source of protein. Let's just say, for example, a pea protein versus a egg protein. The animals on the egg protein are going to tend to outgrow those on the pea protein because one of the essential amino acids in the pea might be compromising growth in the pea protein. So if you analyze the essential amino acids and you find one that's significantly lower, this has O protein, than the one in the egg, this could be the essential amino acid that's compromising growth. Could the same thing happen in people? Absolutely. So if you look at, unfortunately, some of the really malnourished countries in the world, especially those controlled by dictators that are affecting protein sources of kids, if you look at some of these countries, like they say North Korean population in the last generation has been reduced in stature by like 1.5 inches because of compromised protein quality for what's available. So it's very important for kids to have adequate protein during that growth phase. I'm gonna,
There's like a homework assignment for my other classes about like countries and their malnutrition or overnutrition, and 70% of North Koreans are food insecure.
yeah, it's awful. Yeah, I'm surprised it's not higher.
Yeah.
Yeah. Thank you. Okay, guys, thank you. I'll see you soon.